Take one precast polyacrylamide ready gel. This is fragile and contains toxic acrylamide, so you should handle it with gloves and with care. Open the packet by cutting or tearing along the edge as shown. Take the gel out carefully, but do not remove the green comb as yet. Before removing the comb, you must remove the strip of green tape at the base of the gel. Remove the green tape by gently peeling it away from the base of the gel as shown. This will allow the electrical current to pass through the gel during electrophoresis. Now carefully remove the comb. Assemble the gel into the clamp. 2 gels are required for each clamp. Make sure that the gels are the right way up, wells upward, and the right way around, short plate inward, long plate outward. Secure the gels in place carefully by gently pushing the 2 green clamps into position. You should be able to see the wells at the top of the gel if you look closely. At this point, a demonstrator will show you how to rinse the wells. Now you need to place the 2 clamped gels into the gel tank. Note the positive anode terminal, which is red, and the negative cathode, which is black. Make sure you insert the clamp gels such that the black and the red terminals match. Next, fill the inner buffer reservoir with running buffer. Make sure you add sufficient buffer to submerge and fill the wells. Fill to within 0.5 to 1 centimetre of the top of the reservoir. At this point, it is a good idea to check for leaks. Next, fill the outer buffer reservoir to above the 2 gel mark. Do so carefully to avoid air bubbles along the bottom edge of the gels. Now you are ready to load your samples. To load your samples, you need to use a micropipette equipped with a long, fine pipette tip. One sample will be a commercial pre-stained molecular weight marker. You need steady hands and you must watch the sample fill the well as you deliver each sample to the appropriate well. Make sure you know which sample is going into which well. Make a note of this. Do what you can to avoid ejecting air bubbles into the well. This will disturb the sample and interfere with electrophoresis. Be sure not to insert your pipette tip too far into the wells. You must be careful not to damage the well or inject samples directly into the acrylamide. Each of you should try loading at least 1 sample. Once both gels have been loaded, place the lid on the electrophoresis cell. Only one orientation is possible. Once firmly in place, attach the leads to the power pack. Make sure the terminals have the correct orientation. Make sure that your hands and the electrophoresis area are dry. Then, switch on the power pack. Once set, press run. You should see bubbles emerge from the electrodes. You will be able to see the bromophenol blue dye front move from the top to the bottom of the gel. You should also be able to see the pre-stained molecular weight markers separate as the electrophoresis progresses. The proteins in the other samples are not visible, but nevertheless are being separated according to mass in the same way as the pre-stained molecular weight marker. Of course, this video is speeded up. In real time, the gel will take around 45 minutes to run.